Hello, I'm Sue Romanoff. I'm here with the Volition team, and we're excited to share some of their developments in early cancer detection screening. The Volition team's prepared a video presentation, and we'll share that with you shortly. It'll be followed up with Q&A with Louise Baxler, Group Chief Marketing and Communications Officer, Jake Micallef, Chief Scientific Officer, and Dr. Andrew Retter, a leading UK-based hematologist and medical consultant to Volition. If you have any questions during the presentation, please feel free to submit them through the Q&A tab at the bottom of your Zoom panel. We'll try to address them all. Without further ado, let's get started.
Well, good morning and good afternoon. I'm Gael Forterre, the Chief Commercial Officer of Volition. It's a pleasure to welcome you to this webinar today and to introduce you to Dr. Jake Micallef, our Chief Scientific Officer, and Dr. Andrew Retter for their presentations on the breakthrough cancer detection method. Before the detailed science, a few words. Our mission is to save lives and improve outcomes for millions of people and animals worldwide. To do so, we're focused on developing a simple, easy to use, cost-effective blood test to help diagnose and monitor a range of life-altering disease, including cancer, or original focus. Finding cancer, and especially finding it early, has often been described as looking for a needle in a haystack. And indeed, as Jake will explain, it is a needle in a haystack when the needle is also made of hay. The new method presented at ESMO earlier this week demonstrates we have a way to find the needle. This is an incredible breakthrough, and I know that you will be interested in the presentation from both Jake and Andy. The market opportunity is big. It is very big, in the billions. It is bigger than us, which leads us to a commercial strategy. We will license this technology early and seek upfront payments, milestone payments, royalties, and sales of key components. While the data presented is literally off the press, we're already in discussions with interested parties. With that said, I'll keep it there for the commercial part. I will now pass over to Jake and Andy to share more details. Thank you.
Now you've heard from Gael about the commercial opportunities, I'm going to tell you a little bit about the science behind our breakthrough in cancer detection. First of all, a little bit of background. On this slide, you can see the structure of chromatin, and we've shown this picture many times before. It's a string of nucleosomes held together by a string of DNA. But what we've added to it in this picture is a complex called CTCF. And CTCF is a transcription factor that also binds to DNA like a nucleosome, but has completely different functions. CTCF is actually a very important protein that regulates what proteins are made in the cell and actually regulates the structure of chromosomes within the nucleus. There are many big hospitals and quite a few companies that are involved in performing liquid biopsy ctDNA assays for circulating tumor DNA. The challenge that these companies and hospitals face is that most of the DNA circulating in a person's blood, even if they have cancer, is normal, especially at early stage cancer. In stage 1 cancer, for example, it's likely that only 1 part in 1,000 or 1 part in 10,000 of the DNA that's circulating comes from the tumor. The rest is normal. This really is a needle in a haystack problem. And what makes the problem even more difficult is that the needle is really made of hay, because the tumor DNA is not chemically any different from any of the normal DNA. And of course, because it's in the same person, it has a near identical sequence. So how do current ctDNA assays work? They've all really got the same mechanism and the same method, and it hasn't actually changed since the inception of liquid biopsy 10 or 15 years ago. What happens is that you take a blood sample, you extract all the DNA from that blood sample, that is the normal DNA and the cancer DNA, if there is any. You then amplify that DNA to make more, and you do what's called a library preparation, which is basically getting the DNA ready for next generation sequencing. You then do the next generation sequencing. That's a very expensive part. But even when you've got all the sequence data for all of the DNA that was present in that blood sample, it's still mixed up. It's still only 1 part in 1,000 or 1 part in 10,000 from the cancer. The problem's very difficult, and so it's solved by putting the whole sequence data into a big computer program which have very sophisticated bioinformatic algorithms to decide whether or not any of that DNA actually did come from a tumor or not. And over the years, the results for liquid biopsy have improved. They've improved a lot. And the reason for that is that each of those stages has actually improved. So the bioinformatics is better, the sequencing is better, the library preparations are better, and so on. But the basic method is unchanged in a decade or more. The reason the basic method for liquid biopsy is unchanged is because there isn't really anything you can do with DNA other than sequence it. The real answer would be to physically separate the DNA that comes from the cancer, and then detecting it would be as simple as a COVID test. A COVID is a PCR test, and this would be a PCR test. That would make for a very simple ctDNA test. As the acronym says, let's keep it simple, stupid. Having said that it would be good to separate tumor-derived DNA from normal background DNA, of course, it's impossible to do that chemically. However, DNA per se is not what circulates in the bloodstream. What circulates is pieces of chromosomes. So the DNA circulates as protein DNA complexes. Mostly those are nucleosomes, but there are many others as well. One of the others, as you can see on this slide, is CTCF. What we have found is that there are combinations of DNA sequences that occur on CTCF proteins only in cancer. So if you separate out all of the CTCF DNA fragments and then do a PCR, you will detect whether or not any of the cancer combination involving both CTCF and a particular sequence is present. And that will tell you if a person has cancer. What this technology allows us to do is a completely new method for the analysis of ctDNA. For the first time ever, the sequence of events is not simply to extract all the DNA, amplify, library prep, sequencing, and bioinformatics. Instead, we will pull out all of the CTCF molecules and their associated DNA fragments and do a simple PCR test to identify whether or not any of them come from cancer. Scientifically, what makes that possible is really quite simple. CTCF binds to something like 60, 70, or even 100,000 places in the human genome, and it binds to different places when you have cancer. So when a cell becomes cancerous, CTCF binding is lost in some places, but it's also gained in many places. And so where there is a gain of occupancy in cancer, those sequences will not occur on CTCF in a healthy person, but they will occur in a person with cancer. And in a healthy person instead, but all those sequences will occur on nucleosomes. So what does that look like in practice? On this slide, we've got nucleosomes and CTCF molecules, and they are covered by DNA molecules, and most of those are healthy. We have some that are covered by the yellow DNA molecules, which contain the CTCF binding sequence. A lot of those will be on nucleosomes, but in cancer, some of them will also be on CTCF molecules, and you can see that in this picture. Using the occupancy properties of CTCF, we can select sequences that only occur on CTCF in cancer patients. And in this slide, you can see that in a healthy person, the yellow DNA sequence, which represents the cancer-associated sequence of interest, occurs on nucleosomes, the green circle. In a person with cancer, They also have nucleosomes with yellow cancer-associated DNA attached, but in addition, they have the yellow sequence attached to CTCF, and that does not occur in the healthy persons. But if we can remove all of the nucleosomes by separating out the CTCF, then we're left with this picture. So now we have a mixture of CTCF proteins bound to DNA sequences, some of which are yellow and come from the cancer, and some of which are still healthy. Now we can extract the DNA, so we're left with just these pieces of DNA and the CTCF is gone. If you have cancer, you would have the yellow sequences. If you're healthy, you would have no yellow sequences because they would only have occurred on nucleosomes and they would wash away. So the presence of the yellow DNA sequence is an indicator that you have cancer. And we can test for this by a simple PCR, which is like a COVID test. What this means is that for the first time ever, we can physically isolate ctDNA cancer-associated fragments from the normal DNA sequences that contain the same sequence. There's an overall picture here of the method. We take a plasma sample and we add an anti-CTCF antibody to that. That's the purple Y-shaped thing. It pulls the CTCF out of the plasma. We then wash all of the nucleosomes away and we're left with an antibody bound to CTCF. We then extract the DNA from that CTCF antibody and we do a simple PCR test to see whether or not the yellow cancer-associated DNA fragment is present. In fact, one of the beauties of this technology is that biomarker discovery is rapid and very straightforward. So what we did was to take some healthy blood samples. Some samples were from people with other diseases like inflammatory diseases, and some blood samples from people with cancer. We then did the same procedure as before. We pulled out the CTCF associated DNA fragments, but this time we did sequence them for biomarker discovery. So what we did was to see what sequences were present in the cancer samples that were not present in the other samples. And we found 29 sequences that met our criteria. We completed it fairly recently, and then we ran a very preliminary clinical proof of concept. As I said before, we found 29 biomarkers that were interesting. We actually developed PCR tests for 10 of those, and now I'm going to show you just how well those 10 tests performed in reality. Some biomarkers were quite specific for leukemia, but most of the biomarkers were also effective for the identification of a variety of common solid cancers. And different biomarkers were effective for the detection of different combinations of solid cancers, including at early stage cancer. This means it's possible that we will be able to produce assays that can detect particular cancers, for example, a breast cancer test, a prostate cancer test, and so on. And we're currently developing biomarkers for the common solid cancers, including breast, prostate, colorectal cancer, lung, and liver cancer. As I said earlier, biomarker discovery is rapid and low cost, so this won't take us a great deal of time. One point I would like to reiterate is that CTCF has something up to 100,000 or so binding site loci in the human genome, and this represents a potential whole new class of hitherto unappreciated biomarkers that we can use for the detection of cancer. How does it work in practice with real patient samples? So first of all, just using one single PCR test, so now it really is like a COVID test, we were able to detect 61% of leukemia cases with 98% specificity, so really quite high specificity. And you can see that in the ROC curve here, and you can see it pictured with the actual results on the right, where pink is a positive. So we've got one false positive amongst the healthy samples on the left, but lots of pink true positives amongst the leukemia samples on the right. One thing I do want to emphasize is that this is exactly the same as a glucose or a urea or a cholesterol test. There's a simple cutoff. If you're below the cutoff, you're negative, and if you're above the cutoff, you're positive. You don't need a computer. If we now add a second PCR, so we've got a panel of 2 PCR tests, then the sensitivity goes up to 74% of leukemia cases detected, with a very slightly lower specificity at 96%. Most of the PCR tests also detected different solid cancers, and there were 2 or 3 of the assays that were particularly good for detection of colorectal cancer. And in fact, if you put those 2 assays together, we can make a 2 PCR panel that detected 77% of colorectal cancer cases at 92% specificity. And that represents the black dot on this diagram. So what we've achieved here is a blood test that gives nearly as good results as a fecal test, but of course it's much easier to use. And that wasn't even developed on CRC. It was developed for leukemia. We're now developing individual biomarkers that are specific for CRC, and we expect to get results that exceed those that you can get with FIT. And of course, we're developing other tests for lung, prostate, breast, and so on. There's a lot of talk in the liquid biopsy field about multi-cancer early detection tests. So we put another panel together of 2 PCRs that were not specific for any particular cancer or specific for different ones, so that we could combine detection of multiple cancers. And we looked at colorectal, breast, prostate, and liver cancer. We've got patients for 10 of each, except for CRC, we had 13. And we've got positive results of between 50 and 70% for the different cancers. However, what I really want to highlight is cancer stages, of course, because detection of a stage 4 cancer doesn't really help the patient a great deal. What would really make the difference is telling a person that detecting a person's cancer when it's at stage 1 and stage 2, and something can be really done to help them. So we did detect a lot of stage 4 cancers, 75% of them, but the really astonishing thing is that we can detect between 1/3 and 1/2 of stage 1 and 2 cancers. And that really is something I think that nobody has achieved before, to detect early stage cancer in a simple blood test that is quick and low cost. Finally, I'd like to reiterate the advantages of physically separating tumor DNA from normal DNA, because it leads to a much simpler, lower cost, better test. First of all, the cost of traditional NGS assays are $1,000 or more, whereas this is going to cost much less, probably in the region of $100 or so. The traditional assays are very complex, they're not automated, whereas this can be automated. I think the big IVD firms can make a machine that will do this at the press of a button. Traditional NGS is really suitable for high-tech labs. The NHS in England, for example, has 7 labs that perform all of the tests for the whole country, whereas this assay can be done in any central hospital lab, especially if it's automated. It's a lot faster, it takes less than a day, even if it's manual, whereas traditional assays take days or weeks. And of course, there's no library preparation, there's no sequencing, there's no computer involved, and the answer is a simple plus-minus answer, like a COVID test. You don't need a computer to analyze the result. What's coming next? Well, first of all, we will be publishing a paper on this method, including all the details of how to do it and so on. And then we will be next developing particular tests for breast cancer, prostate cancer, lung, colorectal, and so on. And details of those will be coming out over the next few months. As Gael said at the beginning, the mission of our company is to produce life-saving diagnostic tests. And that's also my personal passion, especially if they can be low cost and accessible to everybody, not just accessible in rich, developed countries. Having said that, I'd like to pass you over to somebody that actually understands medicine, and that's Dr. Andrew Retter.
Hello everybody, I'm Dr. Retter. I am a consultant to Volition, and I'm very excited to take you through this talk on cancer today. And thank you to Jake for talking just now. Just to be clear, I'm a consultant to Volition. I do work in the National Health Service, but for clarity, the views expressed here are my own and don't represent those of the NHS Trust where I work. Cancer is the second leading cause of death worldwide. Over the next 20 years, we expect the rates of cancer to go up by about 1.5 times, leaving a huge burden of disease in the population. 5-year survival rates are improving for cancer, which is a brilliant thing. And there's a huge amount of research and the field is rapidly growing and changing. But about 45% of cancers in the UK are still diagnosed in a late stage, in stage 3 and stage 4, where treatment options are more limited. The treatments tend to be much more invasive, much more prolonged, and indeed perhaps even more severe and more difficult for patients to tolerate. And many patients will be older and just too frail to tolerate the intensity of those therapies. So there's a key critical need to improve the diagnosis of cancer and the stages at which we can diagnose cancer, so everyone understands that the earlier you treat something, the easier it is to treat, to interdict, to change its course, and hopefully cure more people. How might the new technology from Volition help? It's aligned with Volition's key principles of diagnostic enrichment. This is using and leveraging modern pioneering biotechnology to improve early stage diagnosis, to monitor diseases and to review their progression going forward. If we can diagnose patients earlier and monitor their treatment more efficiently, perhaps reducing the need for expensive, costly CT scans and MRI scans, or even positron emission scans too, hopefully we can lead to faster, more rapid diagnosis and identification of problems or progression when they occur. What's coming next in this area? Well, we're already in the advanced stages of finishing our first paper discussing transcription factors and their applicability in a large number of cancers. We're starting to develop specific biomarkers for lung, colorectal cancer, prostate cancer, breast cancer, and liver cancers. And together, those are the most common malignancies. Abstracts and more posters are going to be presented at a number of specific or cancer-specific conferences throughout 2024. We have ongoing work with a number of key opinion leaders from centres of excellence to accelerate and progress that research further. So in summary, we're extremely excited to announce this novel breakthrough technology, and we're extremely excited by the transformative effect it could bring to so many patients. It has huge potential of significant clinical and commercial benefit, and it contains a number of truly world firsts. Thank you very much for listening, and I'm delighted to hand you back to Sue. Thank you.
Thank you to the Volition team for that informative presentation and your effort on early cancer detection. Just as a reminder, if you have any questions, please submit them via your Q&A tab at the bottom of your Zoom menu. I'll read them as they come across. Here, for the first question here, thank you for taking my question and for a lot of the interesting presentation. I wanted to ask what was the reaction at ESMO?
Oh, yeah, we were at ESMO last week. We spoke to a lot of people from different institutions and from a lot of companies, and yeah, the response was very good, very high degree of interest, and for most of these people, it was the first time they'd ever seen anything to do with this technology, and yeah, I was very happy with the response. couldn't have been much better, really.
I think you describe it as overwhelmingly positive. It's very exciting, as you've seen from the video just now. It has huge potential.
Right. How about the second one? It says, how does the accuracy of this compare to NGS-based tests?
I think it's early to give a very detailed answer to that. What I would say about that is that in this first embodiment of what we've been doing, that was developed on a leukemia model, and still it gave very good results in all of the solid cancers we looked at, including a very good result at early stage. So I'm confident that the products, when we get that far, will be sensitive to early stage detection, and that the biomarkers that we can develop for each of the individual cancers will be significantly better than the biomarkers that we have by translating what we could find in leukemia into solid cancers. So I think giving you an exact number now was very difficult. We're confident that the final numbers will be good and better than we can do now.
Okay, so we have this other one from Bruce Jackson of Benchmark. Here it says, the CTCF associated with DNA Micallef, are you using the standard DNA extraction methods, or what is the blood sample size of each patient? How does this compare to liquid biopsies?
So at the moment, we are, as I said, we're at an early stage. It's only earlier this year that we've really obtained proof of concept. Currently, we use 1 ml of blood, so it's not a big sample size. It's perfectly manageable. We will be optimizing that, so it might be a bit more or a bit less, but it's going to be around that mark. The DNA extraction is a completely standard extraction kit. There's nothing fancy or difficult about it.
Here's another one. I think this is a follow-up to a prior question. How does this technology differ from NGS tests that are currently available and in development?
I think it's different at a very fundamental level. So the way I see it is that most people involved in liquid biopsy, where it relates to ctDNA, are really focused, or have always been focused on DNA. We as a company have never focused on DNA. We have always focused on the fact that what circulates is fragments of chromosomes. And over the past 15 years as a company, we've been developing immunochemical, immunoassay and others, techniques around isolating and analyzing chromosome fragments. And really, it's that expertise with the immunochemistry of chromosome fragments that has allowed us to make this advance. Because I think it's a fundamentally different perspective to think about DNA. So if what you do is you get a blood sample and you extract all the DNA, in effect you're throwing away the protein component of chromatin as packaging. This isn't interesting, this is packaging. The DNA is interesting and we're going to sequence it. But as a company, we've always focused on the fact that the protein component of chromatin also contains a huge amount of information, predominantly epigenetic. And that can be used if you can get the chemistry of chromatin right, and that's what we've been doing for 15 years now, and that's, I think, what we've managed to achieve here.
So we have a few on the same about how do you protect the IP here?
We've protected the fundamental method in a previous patent 2 or 3 years ago now, and we've also protected these exact methods more distinctly in a more recent filing. So we have obviously we have a huge experience in IP, and we have very good IP people in the company. So I'm very confident that it's well protected. We've also protected it from the perspective of biomarkers. So one of the things, as well as the method, I think mentioned briefly in the video, that this really does open up a whole new arena of biomarkers that nobody's looked at before. Essentially, any differentially occupied, any genome locus that's differentially occupied in cancer by CTCF is a novel biomarker through this technique, and we're protecting that aspect as well.
So we have another question here from Ross from Cantor. As you mentioned, in developing individual biomarkers specific to CRC expected in the first quarter of 24, what is the timing referring to at the beginning of the development of the test or start of the study? When can we expect details on the clinical trial, and how many patients do you think you need to enroll to support regulatory approval?
So for Q1, what we're really talking about here is proof of concept for each of the individual cancers. So it won't be a huge number of patients. It will be sufficient to get a very good idea, a lot bigger than the cohort that we've had to date. But it's going to be 50 to 200 size. It's not going to be 1,000 size, and it won't be sufficient for regulatory approval. What we will achieve by Q1 next year is that we will demonstrate the actual results using biomarkers developed for each of those cancers. What is the sort of result that we're going to get in those cancers in your patient samples, but it won't be regulatory at that stage.
We've got proof of concept, and we're refining that, and we're expanding the number of cancers we can use it in. And as going back to the video, as Gail and Jake said, we're sharing this early because we think it's got so much potential. And really, by showing this proof of concept and developing that further, we really hope to work with others to take it forward and bring it to a huge audience. There are literally millions, if not hundreds of millions of patients this is a potentially applicable technology for. And really, to pick up Jake's comment from the video as well, this is the key here is the separation of tumor DNA and the chemistry that enables that, and that is a game changer, and will bring this test available to a huge number of patients, whereas in many real world settings, next generation sequencing is just not affordable. $1,000, $1,500 test is not, you just exclude a huge number of people, whereas $100 test or something around $100, your market is much, much, much broader. And the other thing we haven't picked up on is you're picking up stage 1, stage 2 disease. So potentially your treatments are less expensive and less invasive at that time as well. So it's one of the reasons we're sharing it early. It completely aligns with Volition's philosophy as a company, and I just think it's tremendously exciting.
Yeah, that's a good transition here, Andy. I have another one for you from Michael Okuthiff. Sorry, apologies for screwing up your name here. From Maxim, how does this fit strategically compared to the Nu-Q nucleosome-based human cancer program? Is this an evolution of those, or a separate program that could be advanced in parallel?
So I'll go first, and then Jake can add in. To be honest, I think Jake's already spoken to this, and it comes from Volition's expertise in understanding how or what cell-free DNA actually means. The literature is a little confusing, and people use the term, but it probably isn't cell-free DNA. You've got DNA attached to chromatin, to nucleosomes, etc., and that's how it's circulating. And really, it's our expertise in that area and the expertise we've developed with Nu-Q that's enabled us to do that. This, as we see it at the moment, this is very much a diagnostic test, which is extremely interesting. Potentially Nu-Q will be more of a monitoring test later, but it's our understanding and experience with nucleosomes that's enabled us to make this next step forward, so to speak. So I see it very much as sort of a sister branch moving forward. But Jake's been doing this longer than I have. See what your thoughts are.
Yeah, it's certainly not a replacement, and we are absolutely continuing with Nu-Q. Incidentally, at ESMO, we had 3 presentations, not one. One of the other 2 presentations was about a Nu-Q test that we've been using for the investigation of nodules discovered on low-dose CT lung cancer screening. And one of the big issues in low-dose CT lung cancer screening is that if you find a small nodule in somebody's lung, essentially you're taking a photograph. Because there's a very small lump in your lung doesn't mean it's cancer any more than a lump that you can feel in your breast may or may not be a cancer. And so we've been working with National University in Taiwan to use Nu-Q as a method of differentiating between nodules which are malignant and benign. And that research has been going very, very well. We have another program in lung cancer with the University of Lyon that's looking at finding minimal residual disease after surgery by Nu-Q. That's also going very well. So Nu-Q is also progressing really, really well. It's just that in this particular webinar, we're talking about CTCF.
Great. So we have another one here from the Freedom analyst. Congrats on the encouraging results. The question is related to early stage cancer samples. Could you comment on the sensitivity rate in this group of patients? Do you see a way of improving it?
Yeah, so as I said, this first study that we did wasn't really intended to be a clinical study. What it was intended to be really was, okay, we've developed this chemistry that we think is fabulous. That's nice. But what will happen when we actually put it into some real samples? And that was really what we were going for with this study. And the results were very good, as you've seen. The next stage is going to be much more about optimizing the chemistry even further. There are a number of things we can still do. This was really the first prototype, if you like. So I think it will get better. And then we also need to optimize what biomarkers are we using for which cancers. Some of these biomarkers out of the 10 that we developed were very good, appeared to be good pan-cancer markers, but we would also like to have more specific markers for prostate, lung, and so on. As you've seen, we detect early stage cancer well. I think when we develop better biomarkers for each cancer, we may do even better than that. Do you want to say something, Ed?
Yeah, so just to pick up on that, I think we've said a number of times now, we're very much sharing this early, and there are a lot of optimization steps we can do to improve it. This model was initially trained on acute myeloid leukemia, and really it sort of slightly leans in favor of that at the moment. But we found it extremely useful, and you were able to see the results with other cancers as well. And there's lots more, particularly with the PCR, that we can do and biomarkers that we're targeting to improve the sensitivity and specificity of the test. There's a huge amount to do and huge potential here. Again, it's one of the reasons why we're sharing it early. But yes, we very much should be able to improve the sensitivity. It's worth also bearing in mind that it's very difficult to pick up stage 1 disease, generally speaking, and it's already performing very well in this area.
So here's, I think this is, you've kind of covered it, but just for clarification, I think they wanted to know for the data for the PCR targets, are they identified for leukemia and they comment to the CTCF binding targets found in CRCR from mammary and lung cancers.
Yeah, that's exactly right. So we identified 29 targets. We made tests for 10 of those 29 targets. So those 10 tests were using sequence biomarkers selected on a leukemia model. So they were selected on the basis that they work in leukemia, but then, we just tried them in solid cancers. We had no idea whether they would work or not. And in fact, most, but not all of the 10 did work in solid cancers. And the interesting thing is that different markers seem to work well in different solid cancers. And I found that actually to be a very encouraging result, because it means that out of even those first 10 markers, we have some that are fairly specific for leukemia and some that were not specific for leukemia, but also worked in other cancers, and they didn't all work in the same cancers. So it seems at this stage, and it's early, it seems at this stage that there are, at least in the case of leukemia, cancer specific as well as pan-cancer markers. And I find very encouraging for what comes next.
So we'll turn this back to Andy here. I think this is a follow-up, and this is more on the cost advantages. Do you expect that to come primarily be driven from the screening setting, where the healthy individuals at risk of cancer would be tested, while NGS may be used down the line in suspected cancer or as a confirmation?
Yes, certainly at the moment, I would expect it to be screening or early diagnosis that you're using it, that very much you're using it for. And I think it offers, if you're talking about large volumes of tests in a screening nature, you can imagine if you're screening thousands, if not tens of thousands or hundreds of thousands of patients, how cost becomes such a huge factor there in its applicability. It's potentially huge savings for certainly within the UK, within the National Health Service, but obviously this is something that we see going very much globally for healthcare organisations throughout the world, whatever their funding model.
Okay, we have another one based on inquiring about the broad range of mutations. I think they're asking if the tests will cover these mutations, and then if the process is more expensive and time consuming, and what the potential is for the Volition test to be capturing multiple mutations in a single test, given that the test targets CTCF with specific DNA sequences.
There's a slight, so to be clear, we're not targeting mutations in the normal sense of the word. So for example, a BRCA mutation, we're not targeting anything like that. It's a new kind of target, if you like. I don't think people have before searched for occupancy market targets. So this is it's a target where this molecule CTCF binds differentially to a certain sequence in cancer. It's not a mutation per se.
Yeah, I think that's a really important point to emphasize. It helps diagnose cancer. It's not designed to say pick up a P53 mutation. Jake mentioned the BRCA mutations too, and actually that goes back to I think the question before last, where you were talking about then doing you've got a very sensitive test, and then you'll do your more specific test later. But this is helping you on that pathway, because you're detecting your cancer earlier, and then you could do your sequencing for breast cancer, for BRCA mutations, etc., thereafter. But we see this as sort of firing the starting gun early to get you to diagnose patients earlier, and that's its real key potential advantage.
And it may be, if I could add something, it may be that further down the line that we find that different patterns of occupancy for CTCF are are also informative about types of disease or efficacy of different drugs for personalized medicine. These may or may not fall out of what we do in the future, but because nobody's ever really looked, it's a whole new arena of biomarkers which may inform all sorts of things, but that remains to be seen.
So I think it might be interesting to kind of follow up on the technology, how it differs, the NGS test, and then what is really available or in development right now.
For me, the real differences are practical. We're looking to achieve the same end as other liquid biopsy tests. I think that the key differences are in ease of use, simplicity, cost, time, and the potential for automation. So I do think that this test, that the big diagnostics companies, big instrument manufacturers, can make a machine that simply does this without the involvement of a human hand, because the first half of the test, isolating the CTCF, is essentially the same as the first half of any immunoassay. So it's like the first half of an insulin test or fertility test, and the second half of it is PCR, like a COVID test. Both of those can be automated, and I think a machine that does both can be put together.
So for me, that it's really that key bit. It's extracting the cancer DNA and identifying the cancer DNA. Whereas with, if you go back to the traditional methods where you're amplifying all the DNA in the sample, you've got to understand what you're looking for, and you need extremely powerful computers to delineate your signal. And even then, it's very complicated. But you've got to know what you're looking for. This is you've got your sample and you're extracting the tumor DNA, and then you're looking for it. And that's a very different way round of doing things. And it's that identification step which is so key. And the fact that we've been able to distill it down to essentially an immuno-based assay form of doing it, that is transformative. And that's why there's such a potential cost reduction, and its applicability is so broad.
Yeah, and just more as a layman, the way it comes across to me is it seems like there's like a common denominator, and you've kind of gotten to that really quickly, and that's the protected IP portion, and that makes it super interesting.
Yeah, absolutely. And I think Gael articulated the strategy taking this forward. Very, I think it was one of the first things said in his presentation, and that's absolutely key. And the final thing I stress is how this absolutely aligns with Volition's ethos, which is to bring diagnostic tests to a broad market, and it's just hugely applicable and very much should make patients' lives better.
Yeah, that's great. Maybe Lou, do you want to take 6 here? We have, or the 8th question here. Great presentation. Thank you very much. My question relates to how you guys are going to monetize this. Really, what are the next steps?
Sure. Well, actually, that's just following on from what Andy was saying in terms of Gael's initial presentation. So firstly, thank you very much for all of the interest. I'm afraid we're not going to get to all of the questions. We've been trying to rattle through as many as possible, but we will be back in touch with people whose questions we've not been able to answer kind of live in this session now. And equally, please do contact us with any further questions that you might have when we set up some separate comms. But in terms of monetization, then very much our strategy here is to license out. We see that we're in the early stages. We've shared this earlier, as Jake said, but this is bigger than us. It's an amazing breakthrough. It's a fantastic opportunity, and we would like to work with others. And already the interest that we had at ESMO this week was really just fantastic. We've already got a number of meetings on the books to take discussions further, and I think that we'll just see this through licensing, as Gael mentioned in his presentation at the top.
So as Lou said, I think that's all the time we have for today. I know this is a very interesting topic. So if you have any other questions, please reach out to the contact information on the press release. Thank you all for joining us here today.